So our summer reading program begins June 1st, and it's a great way to show support for your local libraries by participating. So we just ask you to keep track of how much you read through June 1st to July 13th. We're trying to get 13 million minutes read statewide. And of course, we have some fun incentive prizes and all sorts of programs for all different ages, just to kind of encourage and celebrate reading in our communities. So it's my great pleasure to introduce Sarah Stein, who is responsible for gathering all these incredible experts and all of these interesting people to come and educate us about their field of expertise.
Thank you, Sarah. All right. Well, again, welcome. On behalf of the Hawaii Alliance for Science, thanks for coming tonight. The Hawaii Alliance for Science is a group where it's part of a global initiative for science-based communication, and we grew out of Cornell Alliance for Science. And the Hawaii Alliance for Science is a group of community members, educators, scientists, and we advocate that science can thrive in Hawaii. So this collaboration with the library has been a great way for us to bring in different speakers who can share their science with us. As a reminder, our next talk is going to be on June 10th, and it's going to feature Valerie Psyche. She is from the Hawaii Public Health Institute, and she is actually the community coordinator for Tobacco-Free Hawaii, and she is going to talk about vaping. I went to her presentation about a month ago, and it was fascinating. Honestly, I didn't know much about vaping, and it is a topic I think everyone needs to know more about. It was really cool. So June 10th, she'll be here for that. But tonight, we're happy to welcome back Dr. Jeff Freelinger for his second presentation in our Science YourLibrary theory. And tonight he's going to talk about the development of canine vaccine for valley fever. So Dr. Jeff Freelinger is trained at UC San Diego, Caltech, University of Michigan. He has held faculty positions at USC, the University of North Carolina, and the University of Arizona, as well as research leads at Medical Research Council Radiobiology Unit Harwell, the Institute for Molecular Medicine at Oxford University, and the National Institute of Allergy and Infectious Diseases. So his career has centered around the immune response to microbes, and we're very happy to have him back for his second presentation. So Jeff, thank you. Last talk I forgot. Thank you.
Okay, so what I want to do today is, last time I talked, I showed way too much data. And Dustin beat me heavily about the head and shoulders. And so I'm showing exactly one data slide tonight. But what I do want to talk about is valley fever, which has a great name, coccidioidomycosis. You all going to remember that when we tested at the end. And I'll talk about the reason we got interested in this, and a fair amount about why you might want to start off making a vaccine for dogs as opposed to a vaccine for people. And we'll go through that. And then I will show you at sort of the last third of the talk, maybe even the last half of the talk, my epiphany when I learned about how to actually make vaccines, which turns out to be like really hard, and not for the reasons that you might think, but for reasons of how you actually get something to market, and what the barriers to entry for any new product are. And this is for an animal vaccine. This is not for a human vaccine. Animal vaccine barriers are much lower than human. So this work has all been a collaborative effort for 4 principal investigators at the Valley Fever Center for Excellence at the University of Arizona, and we'll talk about who those people are in a little bit. So, why would anybody want to make a vaccine for valley fever? I'm sure very few people in Hawaii have ever heard of valley fever, much less wanted to make a vaccine for it. So what is it? So it's caused by a fungus. They have great names, Coccidioides mitis and Coccidioides possidacei. We tested on that too, in the spelling of possidacei. It's known by other names, coccidioidosis, people refer to it as coxy familiarly. You want to be familiar with a fungus. It was originally described, about almost 100 years ago, in the medical literature as desert rheumatism. And it presented then when people were getting pretty bad RA, rheumatoid arthritis, like joint disease, and it could be pretty debilitating. And it was unusual in that only inhabitants of the desert got it. People who lived on the coasts, it's essentially never got it. So where does it come from? The infection results from inhaling a spore. So like a small, severity is variable. It ranges from mild, that's about 60% of the people who get infected have mild disease. Moderate disease, that means you're sick enough that you actually go to a doctor. But it also, and then complicated, which can be life-threatening. So that occurs when it leaves, you inhale it, so it goes to your lungs, grows in your lungs, and then sometimes spreads from your lungs. When it spreads from your lungs, that's when it can go to your joints, right? And hence the desert rheumatism, because you get an inflammatory process, which is a perfectly appropriate immune response to the fungus, but it's in your joint, and so it does bad things to your joint. Also can go to your brain, so that really makes you dead. So that's a bad outcome, And after people recover, most of them have lifelong immunity, so they never get it again. Now, the mild disease is really variable, and some people feel sort of crummy for a few weeks. Some people feel like they had mono for 4 months, right? And I mean, they really feel terrible. They'll miss 2, 3, 4 weeks of work. It's not that good. So this is a picture of hyphal growth of the fungus living in the dirt. This is where it hangs out. And
There's a lot of dirt in the desert.
pardon?
There's a lot of dirt.
There is a lot of dirt in the desert, and it's actually pretty widespread. Both the dirt and the fungus, yeah.
Are you going to talk about how the immunity works?
Yeah.
Okay. Then I won't ask a question.
Okay. It spreads, goes in the lungs, and people who don't look at x-rays all the time, this is a consolidated lesion in the lungs. And that's what this turns into in your lung. So this is a spheroblast, and these are a bunch of different about to hatch kind of new fungi that are going to come out. So it's actually pretty interesting. I mean, the life cycle is interesting. People are dead-end host. Actually, most mammals are dead-end hosts, which means it's not contagious or infectious among individuals, so you can't catch it from your next-door neighbor unless you are sniffing the same dirt your next-door neighbor sniffed. But you can't catch it from your next-door neighbor, or your kids, or your dog. So there are probably about 100,000 infections a year, and about 10% of those people actually go try to find a doctor. It's a reportable disease like AIDS, HIV, or gonorrhea, or syphilis. So physicians are supposed to report it to the public health people, but no, not to the CDC, to your local public health people. They're responsible for reporting to the CDC. And there are a couple hundred deaths every year. So not a huge number of deaths, but a lot of cases. And when you take the denominator, which is not the whole country, but is actually the people at risk, we'll talk about who they are. These are the obligate sort of disgusting pictures, so you can see what lesions look. These are cutaneous lesions. I'm told that they're really painful. They look kind of painful, don't they? You know? And you can get these kind of boo-boo-like things, like you get the plague, and you can get them inside in places. So that's one near the spine, which are really painful because they take up space and compress nerves. So, 75% of the diagnosed cases lost greater than 2 weeks of work. 50% had an illness for greater than 6 months. So you're sick for a long time. Right? And there's a really weird thing which we don't understand, and we've worked moderately hard to try and understand, is the risk of having disseminated disease of Filipinos and African Americans is about 5 times that of Caucasians. We spent a fair amount of money trying to look at the genetics of susceptibility by looking at SNPs in African Americans and looking at disseminated African Americans and trying to find particular alleles that looked like they were associated. And we were probably underpowered because we only had about 300 cases, and there's a lot of SNPs to look at. Interestingly, if you run prison systems, I don't know if you care about running prison systems, but in California, there are a lot of prisoners in the jails that are in the Central Valley of California. If California now skin tests for previous immunity every prisoner before they send them to jail in the Central Valley, and only prisoners who are skin test positive, meaning they've already had and recovered from the disease, gets sent to those jails. So if you are skin test negative, you don't go to jail in Bakersfield, right? You go someplace on the coast. There's a court case right now from Hawaii sends prisoners to Arizona to warehouse them because Hawaii doesn't have enough prisons. And there's a court case now from a Filipino, Hawaiian Filipino descent, who's been sent to California prison, an Arizona prison, and is suing that this is cruel and unusual punishment because he's now at risk for getting this disease that he wouldn't be at risk for if he stayed home in Hawaii. So, we talk about humans and dogs. In animals, lots and lots of animals are susceptible, right? And this is the sort of amazing thing. In the Phoenix Zoo, the dolphins got valley fever, right? So dolphins, essentially lots of small mammals, and we think in the wild that small rodents are actually affected, but we don't actually have good evidence for that. Some reptiles are positive. For those who care, llamas and alpacas are also really susceptible, and there are llama and alpaca farms in Arizona, and so it's a veterinary problem for taking care of those animals. So this is where you can find valley fever. The current range is the green, which you can see it goes from South Texas up to a little place in Washington even, right? And so this is all to the east of the mountains. So it's in the desert, all of those places. Thanks to global warming, it's predicted that we're going to expand the range for valley fever. So places which you couldn't get it before, like coastal California, will be able now to have valley fever too. So if you study valley fever, that's actually a good thing, because maybe people will take your then again, maybe we do. Okay, so in animals, this is dogs. About 10% of the dogs every year are infected. So that's an enormously high attack rate, right? I mean, that's really high. There are roughly 400,000 dogs in Phoenix alone, so 40,000 infections. Like humans, 50% are symptomatic, 5 are really sick, 2,000 a year in Arizona. Fluconazole, which is the first line drug, which your dog probably doesn't have health insurance, and so it has no prescription policy. It costs you $500 a month at the pharmacy. We can talk about that because over the last 5 years, fluconazole has gone from about $50 to $500 because there is exactly one manufacturer of fluconazole. It's generic. It's been on patent for 30 years. One company makes it. There are some other azoles that you can use which are even more expensive because they're still on patent. $60 million a year is what it costs for healthcare costs for dogs. 17.5 million dogs in the endemic area. That's a lot of dogs. 10% a year, that's a lot of sick dogs. It's a major driver of economic euthanasia. That's a nice way to say people put down their dog rather than pay for drugs. And the true economic impact is unknown because it's not reportable by veterinarians. So sick dogs don't get reported about healthy people, sick humans. So, incubation time, usually 1 to 4 weeks, so that's bad because then it's really hard to tell when your dog got infected. Because that's a month, potentially away. Disseminated disease can take months to years to show up afterwards. That is, they escape from the lungs, so it's indolent infection for a long time. And the illness is pretty similar to human disease, so that seems to be a little worse for dogs than for people. Okay, so this is a clinical disease, and this is an X-ray of a lesion in the bone of the dog on the bottom right. And you can see that fuzzy thing, that's the lesion. And you can see the dog's lungs on the top radiograph, and it's all dark. It's not, lungs aren't supposed to be dark. They're supposed to be white. So dogs get pretty bad pneumonia. Most common problem things are coughs, weight loss, lymph node, solitary nodules, and they get dogs like people get CNS disease at actually a much higher rate than humans do. So,
What's CANINE?
central nervous system. That goes to your brain, your spinal cord. So, treatment. We just talked about that. Useful drugs, amphotericin B. We call amphotericin B amphoterrible because it's got so many side effects. Rots your liver, kidneys. Not an easy drug to use, not an easy drug to take. Ketoconazole, itraconazole, fluconazole, isoconazole, voriconazole. All expensive, but and all very similar. The other really bad thing about these drugs in valley fever is drugs like tetracycline and bacteria kills the bacteria. The azoles don't kill the fungus. They make the fungus stop growing. So they'll arrest growth, but they won't clear the fungus. So people who respond well and have CNS infections are on drug the rest of their lives. Because as soon as you stop drug, the fungus starts to grow again, and then they get sick again. So bad. So this is the argument about why we have a vaccine. And I want to spend a fair amount of time talking to people, because we don't think of vaccines as made by people. We think of vaccines as made by companies who sort of aren't people. So there are, as I started off to say, there are 4 principal investigators on this project. John Galchiani is an infectious disease physician, and John has spent his entire career taking care of people with valley fever. And he cares incredibly deeply about trying to do something about this disease. He's been at the University of Arizona since about 1970, and he's really dedicated to this. He's now 73. He's still working full time, to try and do this. We got a lot of old farts doing this, right? But it's really important to John to try and push this along. He developed a vaccine, a prototypic vaccine about 20 years ago, a recombinant-based vaccine. And they figured at that time it would cost $50 million to get it through phase 2 trials. Not phase 3 trials, phase 2 trials. Because it wasn't clear it was going to be really expensive to make. And no one wanted to pay for that because they never make their money back. She is the John Galgiani of the veterinarian world in southern Arizona. Lisa is a really excellent clinician, and she has really driven much of the experimental work into my science. So she likes dogs, by the way, in case you couldn't tell, she likes dogs. And she has, last I heard, has 4 dogs, in which she trains for agility trials. Although her Brittany Spaniel, which that is her Brittany Spaniel, isn't so good at that. Sage is the Brittany Spaniel's name. So there are two other people, Mark Orba. Yeah, that would be good. So Mark Orba, notice, School of Plant Sciences. Come on, like work with me here, right? Why would that be there? So what Mark does for a living is he works on pathogens of wheat. And he made a truly inspired guess. So there's a pathogen of wheat which has a gene which attenuates it in wheat. A fungus which is pretty distantly related to coccidioides, but you know, kind of related. And this gene has got a name. It's gene of unknown function. Right? There are many genes of unknown function. After you start to sequence things, you find a lot of genes of unknown function, right? Because you have sequences, you know what they are. But this gene now has a known function that if you knock it out of this fungus and you infect wheat with it, the lesions are much smaller, they don't grow as fast, and the fungus seems sick. So what John convinced Mark he wanted to do, I was not present in those conversations. I don't know how he did this. He needed to start to work on coccidioides and knock out the homologous gene in coccidioides. So coccidioides, when you work on it, is like a pain in the butt. It's a biosafety level 3 organism. So you guys know what that means? That means it has to be in a special room, which has got 1 pass air. Got to be HEPA filtered going out of the room. It has to be, you have to work, you're supposed to work in Tyvek suits with gloves, respirators, because it's an airborne pathogen, right? Because it can go anyplace. So it all has to be in those, working with experimental animals is pain because the animals have to be in these glove boxes. Trust me on this. This is actually not what you want to do in your spare time, because you never can pee, right? Because to pee takes 40 minutes, right? Because you have to go, you have to decontaminate, you have to take everything off, you have to go to the toilet, go back to the toilet, re-gown, re-glove, redo everything, and get back into the lab. Not a good thing. No, well, you drink a lot of coffee after you do. And then me. And so when I first came to UA, I had met John on an NIH review panel a couple of years before when I came. John really sold hard that this was an interesting disease and we should work on this, because we didn't know, still know very little about the immunology. So I'll talk about that for you. So we know a bunch of sort of random factoids. So the immune system's got 3 big compartments: innate, adaptive, and so those. And then the adaptive is split into T cells and B cells. So the innate compartment of things like macrophages, white cells that sort of non-specifically eat things, like the real specific. They make a lot of cytokines which now potentiate the adaptive response. There are T cells and B cells involved. The one thing we know for sure is antibodies are not important. So unlike influenza, where antibodies are really important in resistance disease, makes no difference. We have done, and others have, the experiment where you take essentially essentially all the antibodies out of one mouse and put it into another mouse and nothing else, and ask whether that makes them resistant to the infection. Doesn't make any difference. No effect. We can transfer, so we can transfer B cells, it doesn't matter. If you transfer T cells, you can now transfer resistance. And in fact, there's a flavor of T cells called CD4 T cells that actually are responsible for resistance. So these are the same T cells that get decreased in HIV infection, right? Helper cells. But they actually may do a lot of other things too besides help these cells. They make a lot of molecules, inflammatory cytokines, interferons, they make TNF, that's tumor necrosis factor, all of which are probably important in resisting the fungus, all of which are sort of a knife edge deal, right? Because if you make too much, you cause pathology. If you make too little, you don't get rid of the pathogen. So it's a tricky business to figure out how to regulate those. So John convinced me that we didn't know anything about this, and that anything we figured out would probably be good. Okay, so John sucked into this. And so since about 2012, we've been working on this pretty steadily. Okay, so this is the entire staff of the Valley Fever Center for Excellence, right? So this includes the people, the clinicians, it includes all the scientists, all the technicians, everybody but the janitors. So the first thing you will note is there's not very many of us, right? It's a pretty small number of people who sort of work on a problem like this. And there's John in the back on the left. There are one, two, what? No, there's not 20 physicians. There's like 2 physicians.
No, it's 20.
There's only 2 physicians.
Right.
There's 2 MDs, 2 veterinarians, and 1, 2, 3, 4 PhDs. So not very many people. So when Mark made this attenuated fungus, he predicted that it was going to be attenuated in mice, in people, and anything else. So he made this deletion mutant, and the first thing we did was we put it into mice, like in the same way we always put it into mice. We aerosoled it into their nose and waited around to see if they got sick. So we did it in several strains of inbred mice, and they were all fine. So that was really encouraging, and that we really done something with the fungus. We wanted to know if we took it and put it into a mouse that had no immune system of its own, or at least no adaptive immune system, put it into a mouse called non-skid gamma C null. So these mice don't make interferons, they don't have NK cells, which is an innate cell, they don't have T cells, and they don't have B cells. So we thought, these guys are going to die, right? And in fact, they were fine. Now, unlike normal mice, which cleared that infection with CPS1, these mice didn't clear it. It just kind of hung out for, I think we looked for 3 months. Didn't go away, didn't grow, just kind of was there. So that sort of touches 2 things. One is that the immune system actually does do something. And second, this fungus isn't very bad, right? It's not going to cause serious disease. So this is the only data slide that I'm going to show. So this is an experiment where we took the Delta CPS1, the vaccine strain. We immunized mice. That is, we infected mice twice with this vaccine strain. We waited a month, and we came back with the 2, I told you at the beginning, there are 2 species of coccidiolines. So that's why there are 4 lines on this graph. And we challenged those mice with what would be a lethal dose of the fungus. And you can see that mice who were not been immunized all died, So this is great, right? I mean, it doesn't get better than this if you like vaccines, right? Because we made a vaccine, right? We won. Yeah, we have a vaccine. And, you know, there's some papers that anybody wants to look at them, but what those two papers say is what I just told you, right? You have a vaccine in the works. Okay, so this is when we had our first dose of reality. And, you know, reality is a horrible thing, you know, because it tells you things you thought you understood, but you didn't. And so what we needed to do, and we knew this, is we knew we were incompetent. We knew we did not have to go from this fungus in the bottle to making a vaccine I can put in somebody's dog, right? Because we kind of knew that there were a lot of steps. We didn't know what the steps were. We didn't know how to do them, but we knew they were there. And so we started looking for a partner, you know, that is a company that might be willing to pay for doing at least a big chunk of those steps. At that same time, John and I and Mark and Lisa had written an NIH grant in which we said, oh yeah, well, we have this data and we want to actually figure out how the vaccine works. That was what the grant was written about. So we want to understand the mechanism. That's your question. You know, what parts of the immune response are important? How long do they last? Where do they go? Where do the cells live? All those kinds of things. How long the immune response lasted? You know, how long between vaccinations could you go? So we have some of those answers now. We know that for a mouse, if we vaccinate them, we can wait 9 months, which in a mouse lifetime is a long time, and they're still resistant. We know that it's the T cells that are important. We know that how you give the vaccine matters. We know where you put it matters. But, you know, there's still a lot of stuff we don't know. We don't know which cytokines are important. We don't know how to manipulate those. But we got this grant. Now we had a fair amount of cash. So we started looking again for a partner because we've not found anybody who wanted to pay for the whole thing. We'll talk about one of them earlier. But we found this company, Antibody. So Antibody was a company in Long Beach. And Antibody actually doesn't do anything. Right? Their raison d'etre was to repurpose already known human drugs for animals, of which there are a lot of them. And so their goal was to not actually do any drug development, not do any of those kinds of things, be able to do pretty small studies to show that a drug that works in people will work in dogs or cats or a llama and so or something. But one of the guys on the board, the president actually, was a veterinarian who used to live in Tucson before he moved to Long Beach. And when he was a practitioner, he saw a lot of dogs with valley fever, and he was interested in this. And so he sold to his board the idea that they needed to put up $4.8 million to push this along. And also paid for and recruited a guy to be a project manager. Right? As somebody who's taken multiple kinds of vaccines to one, animal vaccines to one. That was the fundamental turning point for us to be able to drive this process along in the midst of our incredible ignorance about how to do stuff. So this is a slide which I stole from Ed, and so this is how you get over to here, right? Which is like you made something and you can sell it to somebody. This is where we were started, right? From what we had there. We're going to go through this and talk about the my personal opinion, incredibly important and incredibly tedious. So, but there's a lot of them, and if you don't do them, you don't get to the end. So, the first thing you have to do is characterize what you have in the bottle, right? What you want to put in the bottle. So, of course, what we did when we did these experiments, right, is we grew the fungus on a petri dish, and we washed it off with water. We spun it down a couple times, and showed that it was sterile, didn't have anything else in it, and said, "Bob, we're done." Turns out, you can't do that with something you're now going to give to a large number of people, or in this case, dogs. You have to actually do a lot more than that. So the first thing you have to do is figure out how do you harvest it? What do you harvest it in? Right? Water, salt water, buffered salt water, things with antibiotics in them, things without antibiotics. What do you do? And as you start to think about that, there are kind of infinite number of things you could do, right? Because there's kind of no rule. Second question is, whatever I harvested in, how long can I keep it before it goes bad? Right? So this turns out to be really important if you want to have a vaccine that's alive that you're going to deploy. Makes a big difference about whether it has to be cold all the time, frozen all the time, be okay at room temperature, be okay at 37, or in beautiful downtown Phoenix, be okay at 45. That's centigrade. And so, got to do all that. Got to try different ways to harvest it, see how stable it is. Tedious, important. You also then have to, among all the things you test, you have to pick one. So of course, none is perfect. So we had these endless conference calls about how we're going to pick it. What are we going to do? The company let subcontracts in Kansas and Colorado State, and talked to a lot on the phone. The second step is how you actually figure out what to do with it. So it turns out for all living things, you have to make a master seed. That means that's one group of organisms that are split into a whole lot of tubes that you now store, so that everything you use is the same thing. Because evolution happens, right? That is, if you continue to pass microorganisms, they evolve. So what you got after 10 passes is not the same as what you had at time 0. So you have to set up a big chunk of these. And then you have to replicate this at a manufacturing facility, right? Because we can't do that. We're not going to make vaccines. You have to figure out how you move them. I told you this was a biosafety level 3. Not trivial to move biosafety level 3 organisms across the country. It's doable, but you need many permits. You need now to get the people in place who are going to do that. You have to let contracts. To have a contract, you have to tell them what to do, because people won't do stuff unless you tell them what you're going to do, what you want them to do, right? So that's hard, because you don't really know what to do. And then you need to open a project at the Center for Veterinary Biologicals in the Department of Agriculture. So that's the FDA for puppy dogs. Right? So you got to do that. The question is exactly when you do it, and that's not trivial either. So then you have to figure out whether the company can make any money. Right? You have to have plans. You have to meet with the regulatory people about what your plans are, how you're going to proceed to getting this done. Then the CVB has a very firm component that you have to demonstrate that this works. So there are fundamentally 2 ways that you could do this. You could spend a real lot of money and try to do a field trial, like they would do in humans, or you can develop a challenge model, right? That is a way to infect dogs with a vaccine and then show that you can, or the wild type fungus, and then with the vaccine, show that the vaccine works in dogs. So the first problem we had was The last challenge experiment that was done in dogs was done about 1955. So that gave us almost no information about how to do it. The second thing is, turns out, if you want to do an experiment on dogs, dogs are really expensive. So roughly $10,000 to $15,000 per dog to do an experiment. But you can't just, well, first of all, it's not legal and it's not ethical to go around trapping feral dogs to do this. So you have to buy purpose-bred dogs to do this, who have known exposures to pathogens and other microorganisms. They're the dog equivalent of laboratory mice, although much more expensive. And then you have to actually figure out how to do it. So you have to have some way to assay, did you do anything? Right, so we had to develop serology for dogs. We had to develop T-cell assays for dogs. And we had to actually figure out how to do these infections. So that got done at Colorado State. And then you have to go to the manufacturer and tell the manufacturer how to do stuff. So you have to tell the manufacturer how you do stuff. They have to devise some methodology that can actually do it, which is almost certainly different from what you did, which means if they did something different, you have to go back and make sure that what they did didn't change anything. So it's a recursive process, which is goes on for a long time. Then you have to have criteria for what you're going to decide is good enough, right? How viable do the organisms have to be? How potent do they have to be? How long, what's your standard for how long you have to keep? It has to be stable. You have to show that everybody's doing the same thing, right? That means, so I don't know if you guys have ever worked in a research lab, but you know, protocols evolve while people are doing them, even if people are keeping good records. You have to freeze everything. So my father was a specifications and standards engineer, and he once spent a couple of weeks defining what a screw was that went in a missile, right? Because if you didn't define what it was, you know, the screw might fall out, and that would be bad, right? Because then the missile would fall down, and people didn't like that so much, especially the Air Force didn't like that. So those kinds of things are really painful, and you look really sore. So those things are important, and you have to do them. God, are they awful? They're really boring to do. But as I said, important. Then you have to prove that all the stuff that you did, and you can take and you can actually infect a dog, right? And then finally, you have to show that you have some way to read out that you infected the dog, short of dead dogs, right? So we talk about feet up, feet down assays, which is you go into their cage and count the number of paws in the air, divide by 4, and that's the number of dead dogs or mice. But in fact, you have to have some other way, short of that, so we had to make sure the serology worked. So then you have to go and what are you going to do if you actually deploy this? How are you going to tell if it works in the field? How are you going to monitor for bad effects? All of those things, just like you have to do for humans, right? How is all that going to be done? How's the data going to get collected? And then finally, in the end, you get all these pieces of paper and you have to send it to CBP, who has to say it either blesses you or doesn't bless you, or says you have to do something else. And it's unclear because, of course, I haven't done that. And then after you do that, you have to go to every state where you want to use this and repeat the process with the state veterinarians and the public health people for each state individually. So we're back here. This is the process. You know, it's this sort of, and you may have to do this recursively, right? Because if it fails at step 5, you have to go back and repeat things up to step 5. It is incredibly difficult for a fundamental scientist to cope with this, right? Because I told you, I thought we won, right? You know, we had a bug, we could prevent disease, we could all go home, right? You know, we won, we did what we were supposed to do. And it's not even 15% of the effort to run through the whole thing. So, I mean, So if you talk to people about drug development, we were at the point of, in a drug analogy, we had a lead compound, right? We had a chemical that we knew would work, we knew interfere with the process that we were interested in, and we knew it could have a therapeutic effect, right? So that's kind of where we were. If you listen to the drug company guys, that's actually pretty far along, but the cheap part, because I showed you how many people are working on this, right? There's a small number, there are 20 people in the whole center, of which only a few people are actually working on the vaccine, right? So, and the old guys, right, the senior people working on this are working on it 15 or 20% of their time, right? They're doing their day job the rest of the time, you know, teaching medical students, taking care of sick people, teaching undergraduates, seeing sick dogs, you know, all the stuff that you do, which actually earns most of your salary, right? So this is a problem. So I want to spend the last little bit and what it cost and how long did it take and who paid for it, right? So these are not exact numbers. They're mostly pulled out of the sky, but they're not off by orders of magnitude. And the times are pretty close. So we spent 5 years working to actually get the candidate. It's a long time, right? But it's pretty cheap, right? You know, we're under $300,000 a year for 5 people working. That's not 5 full-time people, but it's 5 people working on the project and paying for the mice, paying for all the chemistry stuff that we did, all the media, all that stuff. And it took a long time. Not so long as you think about it. That got paid for almost exclusively by the Valley Fever Center over that time. So that's out of donor contributions, people who thought it was important to try and work on a vaccine for Valley fever. That's all private money. To get to the proof of principle, when we were close, we managed to get a small, short-term NIH grant. And we had for 2 years, in which we could at least have most of those people work, and we were able to get to that experiment I showed you with the live and dead mice, right? And that was the crucial experiment to be able to show that we actually had something. Everything else, you know, was paid for in part by the Valley Fever Center, the National Institutes of Health, and now antibody. And you see that the biggest number there, right, is $5.7 million, which is, is split about 2/3 for antibody and about 1/3 from everything else. So roughly $11 million before we actually had anything to put in a tube and sell to anybody. So we haven't gotten there yet. We're somewhere in the bottom part, but we're working on it hard. I still have 3 people in my lab in Tucson who are going to work every day on this project. And you can see how the number of bodies goes up, the FTEs go up over time. Each of these takes like a lot more people. So we doubled when we started to do formulations, and then we doubled again, you know, when we're really starting to try and figure out how to do this. And there are people all over the US that are involved in this now to try and drive this over the finish line, which it is not yet. So I thought it would be amusing for people to hear this, and I thank you for your attention, and I'm happy to answer any questions. Questions?
Well,
Yeah.
I lived there for 14 years, and I remember it was just assumed you were going to convert your valley fever test.
Mm-hmm.
But I've never heard the numbers like this.
Yeah,
Occasionally you'd meet it was pretty common to hear people, oh, they're feeling bad and stuff. And, you know, we didn't talk about it in such explicit terms like you have. It's just something that, yeah, you'll have it at some point while you're here, so.
So I can tell you 3 of my colleagues got acutely ill with valley fever in the 10 years I've been in Arizona. One of them got disseminated to the CNS, and she had to take early retirement because she could never come back to work, right? 2 others, one of them actually was a graduate student who worked on a different project with us, and he got it, and he also got CNS infection. He responded really well to azoles. They decided he was fine. They stopped it, and he got CNS manifestations for it. So he now is on the East Coast, and he's still on drug. And the 3rd person was one of my close colleagues, and he did exactly what he felt terrible, sicker and sicker over a course of about 3 months. He finally got admitted to the hospital. They misdiagnosed him in the hospital, and it wasn't until John Galgiani went to see him as a visitor, looked at him, and said, "You got valley fever. You be on the wrong drug. You know, you got to be on the right drug." And then he had a big fight with John had a big fight with the attending about who told him John didn't know what he was talking about. The new attending, anyway. He finally got on the right drug, and he started to get better. So it happens, and this guy was really sick. Right? I mean, sick enough to go to the hospital. You have to feel pretty bad before you present yourself, especially somebody who's part of the medical establishment. We have a really high barrier for going to see physicians and going to the hospital because there are a lot of sick people there. There are a lot of sick people there, and you can catch bad stuff there too. So, yeah, but it's, you know, see, I want to skin test Dustin if it was time too in Phoenix, but it's
What a raise.
you're positive. You gotta be positive.
I'm positive you're fine.
So is there any hope that this dog vaccine could have broader application?
Sure, but you know, so the deal is, you know, you're sort of a farmer person, you know, former farmer person, right? You have to pick a thing to get licensed, right? After you get a thing licensed, right? So our hope is that if we can show this works in dogs, we can deploy it in dogs, and it works in dogs, that's actually a major hurdle in being able to get it licensed for humans. And that's what John wants to do. John, you know, I mean, he'd like to cure dogs, but you know, he likes dogs, but he actually has 2 cats. And Lisa wants to cure dogs. Lisa doesn't care about me, but Lisa wants to cure dogs.
But so you chose dogs because maybe that would be an easier
Yeah.
step to adults.
Well, so part of the FDA thing is you need to show efficacy in 2 large animals. So that'd be one of the 2 large animals. So that would be a big step for that, especially a companion animal. And yeah,
Well, you may have answered the question. I mean, this is a path to immunology that I've not heard of using dogs. I understand the mice thing.
yeah. Well, so the dog thing was because there was a clear clinical application, right? And right at 50 minutes, we're good. So I mean, there's a clear clinical application, and that's why we pick dogs. If you're sensible, you would use mini pigs or something because they're way cheaper, right? But because dogs are really expensive, there's a huge ethical barrier to using dogs. People hate doing this. Lisa hated doing it. Hates doing these experiments. I'm not very fond of doing these experiments. And you know, people like puppy dogs. People aren't so keen on mini pigs, but you know, a lot of people like dogs. And we went and we tried pretty hard to get Colorado State to release dogs that were resistant. And the biohazard people would not allow it because they'd been infected with a biosafety level 3 organism. And they would not allow them to be taken out of the facility alive.
So what are the most common large animals? Dogs and
Well, cows, yeah, no, the most common ones are, well, pigs are very common. Primates are primates have a huge barrier. They're incredibly expensive. You know, a 3-monkey experiment would cost you $500,000, right, to do. I mean, it's hugely expensive. 3 monkeys. Monkeys are really really expensive. You have to keep them. If you have to keep them for 6 months, monkey per diems are like $75 a day. You know, it's BSL-3. It's BSL-3 monkeys, right? So it's hugely expensive. Not to mention, it's actually doesn't feel so good to use monkeys, right? You know, it's I worked on SIV for a while, trying to develop an SIV vaccine, and all the monkeys lived in Columbus, Ohio, which I was happy to have them be in Columbus, Ohio. We were in Chapel Hill, and that was fine. And they, as far as we knew, monkeys came in purple top tubes with names on them. That's the anticoagulant. They come in blood draw tubes. And that's all we ever saw. I was happy. I did go visit them once. Not good. Anyway, yeah. Any other questions?
So, excuse me if I didn't, what it sounds like is that how this works is you're infecting them with, in essence, an altered version of.
Right, like measles vaccine. It's like the measles vaccine.
Okay.
It's a debilitated organism that's unable to cause disease by itself, but is able to generate a protective immunity.
Doesn't cause disease at all, or just causes a really mild? No symptoms.
Who knows, right? You know, I can't tell you how the mice feel, right?
Yeah.
You know, they look fine. They run around a cage. They eat. They drink. They don't lose weight. So, a surrogate for disease in mice, particularly, is weight loss. So, you know, we weigh the mice every other day, and if they're sick, they lose weight.
Mm-hmm.
These mice don't. They gain weight. They get fatter. As far as we can tell, they're fine. They're not febrile. You know, you do what you can do, right? But they seem fine. They're clinically well. When you sacrifice them at the end, and you do necropsy, all the organs look fine. Don't look like anything bad's happened. You culture all the bits of a mouse that are convenient to culture that you think might have organisms in them. They don't have any. I don't know what else you can do, right?
So does the organism in the desert require a mammal at some point?
No. In fact, mammals are actually bad for the organism because they don't spread. Doesn't spread. The ecology guys make up stories, right? It's because they have no data, right? So the story they make up is it's a way. What happens is, pack rat gets infected, goes away, dies somewhere else. And now the organism's now got a new place because the pack rat rots, right? And the organism's freed from the pack rat and now it can get out into the environment again. So there's only one problem with that is they've not been able to find pack rats that are infected. So they wave their hands really rapidly, so that's because they die too fast. And so they rot too fast, you know. And so it's a good story, and I like the story. There's almost no data for it.
Well, can the fungus reproduce in the wild without going through a mammal?
Yes. Well, I mean, it grows as hyphae under the sand, right, under the dirt. And you know that's how why dogs get it so much more than people, twice as much more than people, because they stick their nose in the dirt. Dogs probably do that too. I mean, they snuffle all the time.
You can't plant anything without less than that.
Yeah. Oh yeah, no, you know, so people who get it, right, people who are high risk are construction workers.
Yeah.
So there's all these dust mitigation rules that they're supposed to do to keep the dust in. Yeah, you're laughing because you know that.
They weren't doing that when I was there.
Yeah, so they know they're supposed to do that. They're supposed to have trucks spraying water all the time. Yeah. They do it sometimes. Whenever the inspector comes, they're going to truck out there spraying water. There's a great epidemiological set of cases for a bunch of Stanford students doing an archaeologic dig in the desert, an old Native Americans place. There were like 14 kids, 10 of them got infected, right? And so they knew when it happened, right? And so, happens all the time, right? And the other people who get it are veterinarians, right? But they get it because they're out and but those are the people who really get it. And we tell a story when we teach this to medical students. So if you've been in Arizona, you know about the bombs, right? These dust storms. So you say, well, the dust storms, they blow all this dust up and it goes into the city and people breathe because they can't not breathe. And we should see, and we see an increase in valley fever diagnosis 3 months after the homes. And it's kind of not so clear. It's kind of, the epidata is really kind of messy. Epidata is always messy, so that's the story. It's a good story. We tell it because it kind of makes sense. And so for, you're trying to get a medical student to get sort of a sense of what's going on, and that kind of story sort of embeds it in your brain about, oh yeah, this is how that could work, right?
At one point you said, and I don't know if it was an example, you said if you give them tetracycline, it kills the organism.
No, it doesn't kill this organism, it kills bacteria.
Okay.
So, yeah, so it kills bacteria. Tetracycline doesn't do anything to this. So the azoles are the only drugs that have any efficacy, and those drugs are all cytostatic, right? They make the organism not divide. Cytocell.
So in people who've had a mild case and who are now immune to further infection, they are continuing to carry the infection?
No. Well, so that's actually, it's really complicated. I'm gonna lie, right? So one of the ways that this gets diagnosed in people is somebody will go have a routine physical exam. 55-year-old guy will come in and has a physical exam, they look at a chest film, they'll say spot on this. They go, oh shit, this guy's got lung cancer. So somebody will go in and biopsy him, and you wake up and they say, I have great news for you. You don't have lung cancer, but you do have valley fever. So, and there are viable organisms there. So sometimes it can persist. It doesn't seem to do anything. In a way, it's like TB. So 20% of the world's people have TB, and they're fine, right? They had TB, they got better, and nothing happens to them until they get old. You treat them you give them cancer drugs, you get them immunosuppressed, TB comes back. Valley fever will do that too. Get a kidney transplant, your valley fever comes back. So then your choice is to
because of the anti-rejection drugs.
Yeah, because of the anti-rejection drugs. And they inhibit your immune response. All right. Looking for questions.
You got another one?
So, I mean, people with a severe case, what's the treatment? Besides?
Azoles.
Oh,
That's it. That's what we got.
that's all you, okay.
That's what we got. We got 5 or 6 of them. They have slightly different PKs. They have slightly different body distributions.
Were they all cheaper before this latest
Well,
thing
some of them
where
were, because some of them
drug
are newer.
companies?
Some of them are newer, some are still under patent. So they're never cheap. So that's not the first drug you try. But you try, you always try the cheapest drug first. Right.
Yeah.
But still, they'll have to take it for the rest of their life.
Well, they may have to take it for the rest of their lives. If it's not spread to the CNS, it can usually clear it. If you stop it from dividing, the immune system will usually take care of it. But,
So you described this, arduous process to get to the vaccine. Are you in support of this process? Or do you think there should be some shortcuts to get vaccine to market?
I long ago gave up trying to figure out how I would do things if I was God. And I just try and do with what is. And so from my point of view, it's silly to say, well, this is stupid. So I'll tell you one incredibly stupid thing that we're doing that you have to do. One thing you have to do is you have to show that after you give this to an animal, in this case, I think we've talked about it, let's use a mouse. We put it in the mouse's nose. we wait 3 weeks, we grind up its lungs, we take most of it and put it into another mouse the same way, we culture it. We have to do that by law 7 times, right? Even after we can't find anything at passage 2. Because this addresses your problem, right? Does this organism persist? So after you've done it twice and there's no organisms, you have to do it 5 more times. Does that make sense? No. Right? I mean, I did the experiment, there's nothing there, do it some more. You can get overly agitated. You know, for me, when we went through this particular thing, I sputtered for about 15 minutes and said, okay, we'll just do it. We'll just do it, it'll be fine. It doesn't make any sense. As a scientist, it bothers me, but that's okay. I don't make the rules. Yeah.
So are you only shooting to register in Arizona or multiple states, or what's the distribution of valley fever total? I may have missed that.
I showed that slide in the beginning, and it goes.
Oh, okay, yeah, West Coast kind of, how many
Yeah, it
states
goes
is that?
Through, it starts at the in the Sonora Desert, up through Arizona, New Mexico, South Texas, Nevada. Pardon?
There's a spot in Utah.
Yeah,
And
and
a spot in Washington.
a spot in Washington. So, but all through the desert, not the coast, but east of the mountains, right, east of the mountain range.
Does it exist anywhere else in the world?
Yeah, it's in South America.
Same species?
Posadacia.
So are you looking, are you looking to get it registered in all those states then, or are you just shooting for a few, or?
Baby steps.
Yeah.
Arizona first.
Yeah.
Because it's the biggest problem in Arizona, and it's likeliest to get a favorable response. I know that the local community, who's actually given a lot of money to support this, right? I mean, they really want this to happen, and then dog owners want it. You know, we have I didn't talk about it, but we have a pretty big clinical program where we're trying to understand the natural history of the disease in dogs, looking at immune responses and monitoring immune responses in dogs, on and off therapy, and we get more dogs than we can deal with.
At what stage of life would you give the vaccine to the dog?
Well, the answer is I don't know. And we have
That's
no
a good
data.
answer.
And in mice, we give it to young adults. So that's a 6-week-old mouse. Lisa would say she would do it probably at 12 weeks. But she has no reason to say that, except that's when we give a lot of other vaccines. And so, I mean, I think that's just clinical impression, right, without any data. You have to pick something, that's what I would probably pick it up older, but you know, the trade-off there is they can get infected before then, right? And the infection rate's pretty high, but up till 16 weeks, dogs are pretty much inside, right? So the idea is that their chance of getting infected is pretty low if they stay inside. So when they start to go outside and play in the dirt.
And do you know, sorry if I missed this, but do you know whether, how long the immunity lasts?
It only in a mouse. So
You don't know in dogs.
Don't have any dogs.
Yeah.
You know, I mean, you can only do the experiment when you do the experiment, right? And you can't possibly do that experiment in dogs, right? Because we have to keep dogs in BSL-3 for 10 years, 5 years, 2 years? I don't know. I mean, I would get a license before we could actually do that experiment. And the Department of Agriculture won't insist on that experiment. The question is, what do you put on the bottom, right, for the vet? I don't know, we'll make something up, you know.
So when you design experiments, and looking forward to the tremendous amount of oversight, repeating experiments, you talked about do something twice, nothing, do it 5 more times. Does that influence the experiments you design? I mean, that would seem to me to be allow you'd always be dealing with. Let's not do the experiment that way, because that's likely to
Cost us 4 times as much.
raise a lot of questions that don't need to be answered, but there'll be questions anyway, or.
The answer is it depends why we're doing the experiments. So to do mechanistic experiments, we do the experiments that seem to answer the questions that we're interested in, which is, how does it work? You know, what are the cells involved? We do those experiments to do that. You know, we're not doing very many experiments in mice anymore for the companies, right? Because they think they have all the mouse data they need. And we have a lot of mouse data. I mean, we have like 5 years worth of mouse data. So that's kind of, I mean, there's kind of a partition there between trying to understand mechanism and trying to figure out what you need to do for licensure. So the experiments we're doing for the company are things like, how do you tell if you immunize a dog? Right? How do you tell if the dog is vaccinated? Can you tell a vaccinated dog from a dog that's gotten wild type infection? Right? There's actually a name for that, it's called DIVA. And it's one of the things that we've been working on is this because this vaccine's got a deleted gene and we know a lot about what genes are turned down in the vaccine strain. So can we find an antibody that would show up in a wild type infected dog, but not in a vaccinated dog? Right? We don't have the answer to that, but, you know, but that's another thing that we're doing, which is it's kind of interesting, right? But
So do you think there could be a vaccine in 5, 10 years? Any crystal ball is the best guess?
are you a dog? Are you a person?
I'm a dog.
You're a dog, yeah.
We fairly have to do the dog first, so.
Yeah, I think there's a good chance there'll be a vaccine in 5 years for a dog.
So maybe 20 for argument?
I don't know, I'm not part of the FDA. My friends at the FDA, you know, you say live vaccine with the FDA, they go, oh, Jeff.
Do the dogs respond to the skin test they do for the visitors?
No, dogs have crappy skin test responses.